I'm from the Iowa Water Center located at Iowa State University, and I will be your moderator today. The webinar is hosted by the Algal Bloom Action Team, of which Iowa State is a member. The Algal Bloom Action Team is a collaboration of water professionals, researchers, and educators from 12 states in the North Central region of the United States. Team members include the National Network of Water Resources Research Institutes and North Central Region Water Network and University Extension within each state of the North Central region. In addition to hosting a webinar series, our team is developing a website of resources, including fact sheets and frequently asked questions on HABs topics. I encourage each of you to visit our website. I'll put the link in as soon as I'm done with the introduction. And you can explore some of the resources that we have to date, including a what you should know fact sheet, a HABs frequently asked questions database, and recordings from previous webinars and our virtual HABs research symposium, which was held in January. Before we get started, a few housekeeping items. We will have a Q&A session after our speakers present today. So please list your questions and identify which speaker you are posing the question to in the Q&A panel, and we'll do our best to get to all of them following the presentation. If there is a question already posted to the Q&A and you have the same question, you can upvote the question, and we'll be sure to get answers to the most popular questions during the session. Today's webinar is being recorded and will be posted to the Algal Bloom Action Team website following today's session. If you are having any technical issues or have any questions about the Algal Bloom Action Team, please list those in the chat as well, and we will be happy to assist you as best we can. Today's presentation is titled Cyanotoxin Removal by Activated Carbon and Climate Change Impacts on HABs. We will begin with a presentation by Dr. John Lenhart from the Ohio State University. John Lenhart is a professor and associate chair. Sorry, am I still with you guys?
Yes, you are. You cut out there for a second, Laura.
Having a little bit of trouble here with the internet, apparently. Sorry about that. He is also co-director of the Ohio Water Resources Center. He received his PhD in environmental science and engineering from the Colorado School of Mines in 1997. He followed that with a National Research Council postdoctoral fellowship at the United States Geological Survey and a second postdoctoral position at Yale University. In 2003, he joined Ohio State, where he was promoted to associate professor in 2010 and became the co-director of the Ohio Water Resources Center in 2012. In 2019, he was promoted to professor and was appointed to be the associate chair. His research investigates contaminant and particle behavior in heterogeneous systems, with applications to water treatment and contaminant transport in soil and groundwater systems. Current projects involve studying the fate of microplastics in natural and engineered systems, recovering valuable constituents such as critical minerals from industrial and mining waste forms, and elucidating the influence of natural organic matter on the absorption of contaminants to natural and engineered surfaces. He has authored or co-authored more than 100 publications and is the recipient of an NSF Career Award. So now we will begin the presentation, and I will stop sharing my screen.
So it's my turn.
Yes, John, please go ahead.
Okay, is my screen visible?
Looks
Yeah, actually, we actually told James that he was going to be going first. It was just switched on my slides. So
do you want him to go first?
yeah, let's do that.
Okay.
I apologize.
Okay. Sorry, John. All right.
Why don't you let me go ahead and give your introduction as well? I want to just introduce James Cotner. He's a prominent professor in the field of ecology, evolution, and behavior at the University of Minnesota, and specializes in aquatic ecology and biogeochemistry, with a particular focus on freshwater ecosystems. His research revolves around understanding the cycle of carbon, nitrogen, and phosphorus in aquatic environments and ecological implications of these processes. He has conducted extensive research on various topics, including nutrient dynamics in lakes and rivers, the role of microorganisms in nutrient transformations, and the effects of human activities on aquatic ecosystems. His work often involves studying the interactions between organisms and their environment, with an emphasis on understanding the factors that regulate ecosystem functioning. So, all right, James, apologize for the mishap there. Please go ahead.
Okay. Well, it's great to be here with all of you today. The presentation I'm going to give to you today is we're going to talk about climate change and the role of cyanobacteria, both in responding to climate change, but also cyanobacteria as an important driver of climate change processes in freshwater systems.
So Jim, I just want to make sure we're not seeing your slide right now.
Huh. Interesting. So let me try to share again. How about now?
That's good. That's in presentation, or if you get it in presentation mode, we'll be good to go.
All right. How's that? You see it?
I'm only seeing a black screen. Are other people seeing it okay?
Yeah, no, it's just a black screen. I wonder if perhaps it's when you're going on presentation mode that it's getting mixed up here.
Huh.
Perhaps unshare, and then when you share again, just don't go into full presentation mode, Jim, if that might work.
Okay. Well, okay. Let's try that. Okay. Okay, we can just do it like this then.
Yeah.
All right.
Great. Thank you.
Okay, here's a little overview of some of the things I'm going to talk about today. It's pretty straightforward. First, we're going to talk about what climate change is. And I know most of us think we know what that is, but I just want to make sure we're on the same page. Then we're going to talk about harmful algal blooms. And when I'm talking about harmful algal blooms with you folks today, I'm talking about cyanobacteria. And there, the point will be that there are lots of different ways that cyanobacteria are successful in ecosystems, but that also can present a lot of problems. And John is going to tell us today how to solve those problems. I'm mostly really good at identifying the problems in the first place. And then we'll finish up today talking about how climate change is or will enhance the growth of cyanobacteria in the future. And importantly, what the feedbacks of cyanobacteria are back to the climate. And the short story there is that essentially in freshwater systems, it's most likely that cyanobacteria are a positive feedback to climate change. So I guess if I'm in this mode, my animations aren't going to work very well. So first we can ask the question, what is climate change? And as I mentioned, I think most of all of us are aware of some of these things. The most important thing being increasing temperatures. And this figure shows you over the last 2,000 years, basically, our estimates of what the global mean temperature trend has been. And clearly there's an anomaly in the last 100 to 200 years or so. And we think, of course, that that's due to human impacts on the climate system. But there's lots of other things that are associated with climate change that maybe we don't think about so much. One being drought. That's particularly in the upper Midwest right now. That's something that's particularly problematic. Flooding, there's lots of flooding occurring in California this year associated with huge snow events. Sea level rise is certainly problematic in coastal regions. And in general, just increased storm intensity, whether that's hurricane or tornado or thunderstorms or what have you. And one of the things that I like to point out is just, essentially climate change is a phenomenon where we're putting more energy into the climate system. And that more energy means that we get more extremes. And one way to think about that, to me, it's like, as a kid, when you played with a top, you start the top and it spins and it's very stable. And when it has lots of energy associated with it. But then as the energy starts to dissipate, the top wobbles a lot more and eventually it'll fall down. But essentially what we're doing by putting more energy into the climate system, we're emphasizing a lot of these extremes. And that's what leads to these conditions that are problematic. So climate change is not increased CO2 concentrations. Climate is a response to those increased CO2 concentrations. And we know that CO2 concentrations are increasing because we monitor it intensively. The longest record of CO2 measurements have been made at the Mauna Loa Observatory, made famous by Charles Keeling. But basically what they show is that on an annual basis, if you look at this figure, these red dots are going up and down. So CO2 concentrations are higher in the Northern Hemisphere during the winter time when there's less photosynthesis, and the CO2 concentrations are lower in the summertime, as we're going into right now with increased photosynthesis. And what's over here on the right side is basically this same diagram going back to the 1950s when Keeling began these measurements. And as you can see, we're at about 420 parts per million right now. Another important climate change gas is methane. So methane has 30 to 50 times the global warming potential of CO2, but we're also increasing the concentration of methane in the atmosphere, and that's certainly problematic as well. So I'm going to talk a little bit about feedback responses, and essentially this is a cartoon illustrating different kinds of feedback, and it's very simple. So you can have a system where you have a condition on the left that's affecting the system, which is located on the right. And if there's no feedback, basically that means that the B is not having an effect on A. If you have negative feedback, that means that the condition that's imposed by A onto B leads to a deceleration of whatever that condition is. And then a positive feedback means that the response of the system leads to an acceleration of the condition that led to the response in system B. And of course, negative feedbacks attenuate and are stabilizing in a system, whereas positive feedbacks lead to acceleration of processes and are problematic in terms of stability of the system. Okay. Freshwater systems both respond to and are drivers of climate. And the responses are things that you've probably talked about in this series many times. Some of the important responses are things like changes in water levels. So as precipitation increases, water levels go up, or vice versa as precipitation decreases. Changes in nutrient concentrations are really important in terms of climate, because as precipitation increases, it brings more nutrients into a system. And those nutrients basically lead to changes in the behavior of that system, and vice versa as droughts become important. Another important response related to climate are changes in salinity of a system. We don't think about salinity so much in freshwater systems, but certainly it can be problematic in systems like the Great Salt Lake that's drying up right now. And that's going to have effects on the food web and the different species that are found in those systems. But mostly in the things I'm talking with you about today have to do with that last thing that is starred, which are changes in the food web. And specifically changes in the food web that are related or due to cyanobacteria. But as I mentioned, freshwaters are also important drivers of climate change. And this is something that we didn't really appreciate until about 15 to 20 years ago. Freshwater systems both release, they release a lot of both CO2 and methane into the atmosphere. And as we've already discussed, CO2 and methane are important drivers of climate. So specifically, when we talk about CO2, freshwaters release about 4 petagrams of carbon dioxide into the atmosphere per year, okay? Just so I make sure we're on the same page, a petagram is 1 times 10 to the 15th grams. And that probably doesn't mean a whole lot to you, but here's an illustration by Chris Sabine, an oceanographer that studies climate change. He made the analogy that if you took 1 petagram of carbon in the form of coal and put it in a train, a train car full of coal, basically, it would wrap around the Earth at the equator 6 times. And humans are putting 10 petagrams per year of CO2 into the atmosphere per year. But so keep in mind, freshwaters are doing about 40% of that on their own. Okay, so freshwaters are a significant source of CO2 into the atmosphere. They're also, and perhaps even more significant, input of methane into the atmosphere. So freshwaters release about 170 to 200 teragrams of carbon into the atmosphere per year. So quite a bit less, but keep in mind that methane has a warming potential of about 30 to 50 times that of CO2. So it's comparable in warming potential to that of what CO2 is doing. And then to put freshwaters in the context of what humans are doing, we put about 300 20 teragrams per year due to primarily agricultural and industrial processes of methane into the atmosphere per year. So in both respects, freshwater systems are really important in terms of carbon cycling. We can ask the question, though, to paraphrase the late great Tina Turner, what cyanobacteria got to do with it? And if she had sung what cyanobacteria got to do with it, she probably wouldn't have been famous. But nonetheless, we can use that as a launching grounds to talk about what cyanobacteria actually do in freshwater systems. And I made this cartoon to illustrate that. So on the left is a non-cyanobacterial lake, and on the right is a cyanobacteria-dominated lake. And just to be clear, there are, as far as I can tell, there's no lakes that don't have cyanobacteria in them. The difference mostly has to do with whether or not they're the dominant algae that are found in a system. So the cartoon is basically illustrating a lake in the summertime. So it's red at the surface, illustrating that it's warm near the surface because it's warmed by the sun. We say that lakes are stably stratified because the warm temperatures above are less dense than the colder temperatures below. And that leads to stratification. So the algae are up here in the surface, they're photosynthesizing, they're producing oxygen, they're also producing organic matter. That organic matter is consumed by the world's cutest herbivore, which is Daphnia. And Daphnia are consumed by planktivores, such as fish, sunfish maybe are commonly eating Daphnia. And they're consumed by piscivores. In these kinds of systems, there's typically high levels of oxygen because this organic matter in the form of phytoplankton doesn't accumulate. It ends up in Daphnia, it ends up in planktivores, and it ends up in piscivores. Nonetheless, these systems release both CO2 and methane into the atmosphere on average. But in a cyanobacteria-dominated systems, they release a lot more CO2 and a lot more methane. And that's because the cyanobacteria have protections against herbivores. They tend not to be consumed as much. And so more of this algal biomass will accumulate in the deeper parts of the lake or in the shallow sediments where it consumes oxygen. And so these systems tend to have low oxygen levels and truncated food chains, okay? And those factors, because of the low oxygen, and methane is actually produced under low oxygen conditions, more of that methane is released under those conditions. So, as I mentioned, cyanobacteria can be many different things. Cyanobacteria have been around for about 3 billion years on this planet. So they're the oldest oxygenic photosynthesizers on the planet. They've seen a lot of things in Earth's history, so they can tolerate and exploit high temperatures. They're used to stable stratification in systems. And so they can exploit that much better than a lot of their competitors. They can exploit high nutrient and high CO2 levels, which is, of course, is increasingly problematic. And they can also exploit low nutrient levels, which is a condition that's characteristic of our lakes late in the summer when nutrients have been consumed to a significant extent. And when we talk about cyanobacteria, this cartoon is meant to illustrate just how diverse the cyanobacteria really are. They're filaments, they're colonies, they're single-celled organisms. And so they have lots of different habits. Some of them fix nitrogen, but not all of them. Some of them have these heterocysts, which help them do that. So they're incredibly diverse. But the bottom line is they're good competitors when the times are tough. So when nutrient availability has been depleted, they tend to take over in many of these systems. So how do they do that? Well, a lot of the things that we've mentioned already, they can grow fast, especially at high temperature and when nutrient levels are high. They have the capacity to move up and down in the water column, and that's really good at exploiting those stable stratification systems that is occurring in our lakes right now due to the warming that's occurring on the surface of the lake. So they can move up and down, and that's ideal because phytoplankton need to oversimplify two things. They need nutrients and they need light. The nutrients tend to accumulate in the bottom of the lake. So they can migrate down into the water column to pick up nutrients, and they can migrate up in the water column to pick up greater light levels. They're also really good at taking up nutrients and storing them internally. So nitrogen, phosphorus, and carbon they're particularly good at doing that. And lastly, they have lots of protection against predators. So they can form sheaths around them that makes it hard for the predators to eat them. As many of you know, they can make toxins and other adaptations such as spines that makes it difficult for things like Daphnia to eat them. And so consequently, when we look at a typical lake in this part of the world, we see typically a bloom of algae that occurs in the springtime when the lake is mixing all the way to the bottom. And the algae that are prevalent then tend to be diatoms, cryptophytes, and other quite edible plankton that the Daphnia will consume. And so they'll draw those species and those communities down. And then by the time summer sinks in, what happens is we get a bloom of cyanobacteria because they're less edible and they're quite competitive under the lower nutrient conditions that are going to be prevalent at that time of year. So cyanos also, as we mentioned, have feedbacks to not only climate, but also nutrient availability. This is the same diagram I showed you before where we have the truncated food chain, but one of the manifestations of low dissolved oxygen, which is precipitated by these cyanobacteria blooms, is that it leads to the release of nutrients, especially phosphorus, from the sediments. And so now you can see that this is definitely a positive feedback system where more cyanobacteria means more phosphorus released from the sediments, which stimulates more cyanobacteria in future environments. Another way that cyanobacteria lead to positive feedback is those low oxygen conditions. So this figure is a little complicated, but it's from some work we've done in a lake here in Minnesota. But we're looking across the summertime here. This is a stratified lake. You can see oxygen is high in the summertime, especially in the springtime. And this is that spring bloom, that diatom bloom that we saw in that in previous figures. And what happens is this bloom then falls to the bottom, and these low oxygen conditions down here are what are leading to the phosphorus release from the sediments. But it also leads to carbon dioxide increases in the bottom waters. And that CO2 diffuses up to the surface and helps maintain the cyanobacteria bloom in the surface waters. And Lastly, to summarize all this, basically cyanobacteria are a positive feedback to not only climate change, but also nutrient levels in freshwater systems. And the climate change feedback is mostly mediated through CO2 and methane. So climate change, to really oversimplify things, climate change in freshwater systems essentially means more summer. And the summer condition in a lot of freshwater systems is characterized by the bloom, cyanobacteria blooms. And so climate change in essence means that we're more than likely to see more cyanobacteria blooms in the future. And with that, I will thank you, and I guess we'll take questions later.
Very good. Thank you, Jim. That was an excellent presentation and appreciate you sharing that with us. And as he said, we are going to take care of questions after John presents his presentation. And for those of you that were here, I introduced him early on, but I just want to reiterate, next is John Lenhart, and he's going to be talking about factors controlling cyanotoxin removal by activated carbon. So, John, you may go ahead and share your screen.
All righty, good afternoon for me. I guess it's still morning for a lot of folks. Can you see my screen okay? And hear me okay? All good. Okay, well, thank you. So we're going to change gears here quite a bit and talk more specifically about toxin removal during water treatment. And so the research that I'm going to summarize, the impetus was based upon a series of water quality issues that occurred in the Western Lake Erie Basin it was about 10 years ago. So in 2013 and 2014, there were two separate episodes that resulted in do not drink from the municipal water supplies. The one that I think people are most familiar with was in 2014 for Toledo. And that one occurred for 3 days and impacted about 400,000 people. So the consequence of that was a lot of interest in the state of Ohio in trying to assist utilities in modifying their systems, adapting their systems for seasonal algae blooms, particularly on the Lake Erie Basin, which serves so many municipalities. So in Ohio and throughout the Great Lakes, a lot of water utilities that use surface water for sources use what I call conventional water treatment approaches. And this is sort of schematically demonstrated here. And the idea with this is you can take a variable quality source water, such as a HABs impacted water, and through a series of unit processes, produce water that is palatable and potable. And this is an approach that's been applied for the better part of 120 years, and for the most part works very well. And in essence, what we do is early on in the process is we'll add some chemicals. The chemicals primarily are designed to cause the particles that are resident in our source water to stick to one another, so that as the water is slowly churned or agitated, these particles will stick, form big aggregates that we call flocks. Those flocks are then removed during sedimentation, some of them, not all of them, and then subsequent particles are removed during filtration. And then after filtration, additional disinfectant is typically added, provide a residual, prevent any sort of future biological or pathogenic growth. And so this whole process then produces most of our municipal drinking water that we utilize today. So again, what it does is provide for stable water conditions, removes particles, and inactivates pathogens. What it does not do, and is not intended to do, is remove or manage organic contaminants or other trace contaminants that are not sort of traditionally present in our surface water. So things like cyanotoxins would be one of those, and the extracellular form of the cyanotoxin is then particularly problematic for water utilities. So what they do is add activated carbon, and if it's a seasonal issue, they will use powdered activated carbon, and they'll add the powdered activated carbon as early as they can in their treatment approach. That allows then for sufficient time for the powdered activated carbon to interact with the water and remove the toxins. But several years ago, what wasn't known was which carbons to use, under what conditions to use them, how much time is needed to get good removal, how much carbonol is removed. And so this is kind of what we started looking into. And for those of you who aren't familiar with powdered activated carbon, this cartoon schematic shows a cross section of a carbon particle. And what you want to recognize is activated carbon is a very porous material. In fact, most of the surface area of the activated carbon is internal to the particles themselves. So this is what makes them such valuable adsorbents. They have a lot of internal surface area. So the particles are organized, or not organized, they're characterized on the basis of their size. So you can have big particles, we call those macropores, those are greater than 50 nanometers. Meso, or the medium-sized ones, 2 to 50. Micro then are very small, less than 2 nanometers. So the size of the particles within a particular activated carbon and how those pores are connected to one another is very important. And for our purposes, when we're looking at cyanotoxins, what we have to recognize is the toxins have different sizes. So this schematically demonstrates size differences between a microcystin, saxitoxin, and then iodine. And the reason I have iodine on here is because that's a common compound that's used in the industry to sort of demonstrate the available surface for adsorption for a particular carbon, but the iodine molecule is very small, much smaller than saxitoxin, and saxitoxin is much smaller than microcystin. So when we look at the adsorption of these different contaminants to our carbon, the iodine can access a lot more of those interior pore spaces, particularly the micropores, than can the saxitoxin, and much more than the microcystin. So we have to understand then for our particular carbon, what sort of pores does it have, and how will that then affect our interest in terms of removing toxins. Another thing to sort of consider, and I'm not going to talk about this too much, is the carbons themselves have some physical chemical properties that can influence things like charge. So under certain conditions, that's important, but we're not going to talk about that today. So the toxins that I want to talk about that we've studied are two specifically. So we did some work with saxitoxin, and then we've done some work with a variety of microcystins, but what I'll talk about today is specifically microcystin LR. So I think many of you are familiar with the structures of these. What's important to note is the size differences. I already mentioned this. So the microcystin with a molecular weight of about 1,000 grams per mole, depending upon which microcystin you're looking at, is about 3 times larger than the saxitoxin molecule. Another thing to note is the charge. So saxitoxin is positively charged under most of the water conditions that we would be interested in, so pH less than 9. On the other hand, microcystin LR is negatively charged under these same conditions. The other microcystins, some of those are neutrally charged and some of them are more charged than the microcystin LR, but for microcystin LR, pH less than 9, it's a negative charge. Within Ohio, microcystin LR is a very commonly measured microcystin, as is probably the case in most regions. We also had recently seen sporadic measurements of saxitoxin, so it became of interest to the local utilities and to the regulatory agencies. They want to understand, if the saxitoxin were present, how well would the carbon work? It's particularly problematic because the mode of toxicity for saxitoxin, which is a neurotoxin, is much more detrimental than is the mode of toxicity of microcystin. So they were concerned about that. So when we're thinking about using powdered activated carbon, or PAC, PAC, we have to consider 3 things: the carbon properties itself, the properties or characteristics of the toxin, and then basic characteristics of the water. All of these are important in our determinations. So first and foremost, with respect to the PAC properties, are the source itself. So the source then dictates the properties of the carbon and produces carbons that have different physical and chemical properties. And so that's important to recognize. And what's really important to recognize is how these then produce particles that have different amounts of pores or different higher concentrations of porosity and what those pore sizes are. And so that's something we're going to talk about. The other thing then would be with respect to the toxin characteristics. I've already kind of gone through an overview of that. So the idea is, depending upon the toxin you're interested in, its size and charge and also its solubility or hydrophobicity is going to differ. And that's important in terms of how well it might be removed by activated carbon. And then in terms of the water characteristics, what is particularly important to understand is organic matter. Jim already mentioned organic matter, and in terms of water treatment, organic matter is something that's very problematic as it interferes with a lot of water treatment processes. Other factors on the basis of water characteristics also influence removal, things like pH, but we're not going to talk about that. We're going to focus on organic matter. All right, so let's go through a summary of some of the results. And so here I've summarized for our two toxins, the relative removal rate for some different pack materials that we've tested. So I have microcystin LR and saxitoxin, and the relative removal rate is normalized to, in this case, the removal for the microcystin LR to what's called bit coal. So that's an activated carbon that was made out of bituminous coal. And so that we are just sort of setting a removal rate of 1. And so that you can then compare the relative removal rates for the different carbon types or for the different toxin to that value of 1. And so for the microcystin LR, what we can see is that the wood-based carbon removes the microcystin about 10 times faster than does the bituminous coal. So there's a greater dependence on the rate of removal of the microcystin for the different carbons than there is for saxitoxin, because the variation in the removal rates for the different carbons that we looked at for saxitoxin, they only change by about a factor of 2. So what that means is, if you're looking at the time for removal, the contact time, and you have microcystin LR in your system, and you're using a wood-based carbon, it will remove the microcystin 10 times faster than would the bituminous coal. So that's something that, from a utilities perspective, is important to note. And there's a reason why this is, and that's a function of the properties of the carbon. So in a cartoony way, I'm demonstrating here at the bottom how the porous nature of these different carbons vary. So I have a wood carbon, a coal carbon, and then a coconut carbon. So the wood carbon is characterized by having a fair amount of our medium-sized pores. And so those medium-sized pores can handle most toxins. They have a sufficient size so the toxins can penetrate, diffuse into the interior of the carbon where they can be removed. If we go all the way down to the other side, the coconut, so the coconut is characterized by mostly small pores, very small pores. And so what that means is it can only accommodate very small compounds. So in some cases, the saxitoxin can interact with some of those pores. And if we were looking back at the iodine that I talked about previously, it can access those as well, but microcystin cannot. So for example, in the figure, I'm not even showing our results with coconut because the coconut really didn't work at all. So we didn't show that. So that summarizes kind of the carbon properties. Again, it's important to understand the porous nature. So what I'll talk about next then are the toxin properties. And so this is a similar sort of figure, but the parameter I'm comparing is different. So this is a partition coefficient, or a KD. And what you can see in this case, that the KDs, this is a log plot. So the KD for microcystin LR is about a little over 2 orders of magnitude greater than is the KD for saxitoxin. And the KD is sort of a measurement of affinity. What that means is that the microcystin, it has a greater affinity by over 100 times to the carbon than does the saxitoxin. So that means it's going to be removed much more effectively. It likes carbon more than does the saxitoxin. What's important to note also is that in the case of microcystin, we did some work with other microcystin types, and we did see some variation. And the reason why the microcystin is attracted to the carbon more so than the saxitoxin, from our perspective, is mostly based upon the chemical nature of these compounds and their solubility or hydrophobicity. So the microcystin is a larger molecule. It's more hydrophobic than is the saxitoxin. And so that lends it to be attracted to the carbon more than is the saxitoxin. So what I'll talk about next then is water composition, and specifically what we were looking at is organic matter, and we measured organic matter on the basis of total organic carbon. So we have results here at 3 different TOC values. So water without any organic carbon, and then water that has 5 milligrams per liter of organic carbon. So that would span the ranges of TOC values that you would often see. Certainly you'd see higher than 5, but 5 is kind of where we stopped. And again, this is a relative removal rate. So we're looking at how quickly a particular carbon would remove a particular toxin, and this is specific to microcystin. And so what we can see, if we don't have any organic matter in our system, that the wood-based carbon will remove the microcystin 60 times faster than, in this case, the coconut actually got some measurement, 60 times faster than the coconut. And then also quite a bit faster than the coal blend or the bituminous coal. If we have some organic matter in our system, as little as 2.5 milligrams per liter of carbon, we see the removal goes down quite a bit. This is due to competition with the organic matter. Interestingly enough, if we increase the carbon up to 5 milligrams per liter, we don't see a big effect. So there's a sort of a level where the amount of activated carbon and where the amount of organic matter in the system doesn't influence removal very much, because the amount of competition does not necessarily increase as the organic matter content increases. I'm not showing the saxitoxin results here, but just kind of recognize that we saw similar results with the saxitoxin, but they were a bit more muted just because the saxitoxin wasn't removed as well to begin with. And so schematically what's occurring here, so we have our carbon, it's got a variety of different pore spaces, the microcystin and saxitoxin like those pore spaces, and we'll go in there to try to absorb. Our organic matter would be molecules of different size and shape and different sorts of chemical and physical properties. They also like those, and so there's competition. And depending upon the chemical and physical properties of the organic matter, it may penetrate into the pore spaces where it can actually directly compete for adsorption sites, or in some cases it can actually block or occlude pore spaces, preventing the toxin from reaching the interior altogether. So the combination of both of those is what results in the reduction of absorption and the competition. So just to kind of start summarizing, so in general, the pack that had the medium-sized pores, the mesopores, would work best for all the toxins we looked at. Some of the literature suggests coconut-based or carbon for saxitoxin. We did not see that perform very well. Again, the wood-based carbon with the mesopores worked best. We also are looking at testing this for taste and odor compounds, because that's a common problem with a lot of water utilities. And for the most part, for water utilities, the taste and odor compounds are pretty small. And so a coconut-based carbon is something that they would utilize. So we would want to look at some of the other carbons. As I've already indicated, the microcystin is removed much more readily than is the saxitoxin by pack. It just has a greater affinity for the carbon than does the saxitoxin. In terms of the dependence on the pack type, the microcystins exhibit more dependence on the pack type than does the saxitoxin. And so therefore, it's important to note that the toxins that are present, because their properties are going to be influencing the removal, and then the choice of pack would then reflect that. The inhibition of removal for microcystin by organic matter was much greater than it was for saxitoxin. And again, this is a function of the size of the microcystin and its ability to only access a certain types of pores, and then the organic matter interferes more with that. And then last things to consider is kind of the trade-off of cost of carbon versus how much carbon versus time. So the idea would be that you could use a carbon that doesn't necessarily perform as well because it's cheaper, but you would have to use more of it and you would need more contact time. So there's kind of some calculations that need to go into there. In terms of availability, that's also something to consider. The carbons that we used were all sourced locally, commercial carbons. We did not do any special processing. We did not look at any alternative sources. So these are all commercial-based ones, and depending upon your location, you may have access to different ones. From a utility or water plant's perspective, they are very risk averse, and changing up their systems is not something they're prone to do if what they're doing is working, because they don't want to have any sort of issues with respect to producing the water. So the idea of introducing something new requires a lot of consideration and work. So that's something that it has to look at. And then lastly is the issue with residuals management. So the carbon binds the toxins, and then it is removed during sedimentation. So within a water plant, then the carbon is associated with the residuals, and those residuals have to be managed in some manner. And so there's a question as to how the toxins that are associated with the carbon and these residuals impacts that management. Are the toxins released? Are they consumed biologically? So those are all questions that remain somewhat unanswered. So I'd like to acknowledge funding, so a variety of funding sources, including Sea Grant in Ohio, Ohio Department of Higher Education through their harmful algal bloom research initiative. A variety of students, both at the graduate level and undergraduate level, have worked on this, have images of some of those here, and I am now done. So I think we now have time for questions for both of us.
Very good. Thank you so much, John, for that presentation. And yes, we are going to be moving into our question and answer. And I just want to share my screen so that you have information on how to get a hold of our presenters today. And I'd like to thank everyone for participating in the Algal Bloom Action Team's webinar series. Really special thanks to our presenters for sharing their research with us. And thank you to Ann Nardi and Joe Bonnell for their team at North Central Region Water Network for hosting this Zoom webinar and coordinating the technical aspects of the webinar. We couldn't do it without them for sure. So following the Q&A, I'm going to be posting a link to a survey. So please take some time to take a look at that and respond. And so we're now going to move into the Q&A session of the webinar. And please use the Q&A box. You do have an option of upvoting anything that you see as posed as a question that you have so that we can prioritize. So with that, both John and Jim will be getting on with this. So the first question looks like it is for John. Can activated carbon be used at a lake level to improve lake water quality?
So the idea would be to add carbon to a lake. I don't think that's traditionally used because it's going to be a temporary impact. You might remove the toxins over a brief period of time, but you'd have to continue dosing it in order to get removal of those toxins. So it's mostly a point of use sort of approach for removing the toxins during the treatment of water. There might be alternative ways to deal with the toxins in the lake or dealing with the actual organisms themselves that are producing the toxins. Hopefully that answers that question.
Okay, and I've got another question that several people have. It's seemed like cyanos take advantage of competitive edge during a low nutrient summer months. If I got that right, would the implementation of ag BMPs in the watershed be helpful to reducing the occurrence of HABs?
Oh yeah, no question that would help. And I think there was another question that was kind of related to this, so which has to do with, you know, on one hand, we're saying that cyanobacteria are good competitors when nutrient levels are low, yet high nutrient levels promote the growth of cyanobacteria. And I know that that's confusing, but essentially what that means is if you have high nutrient levels that are capable of being consumed by algae that are in the water, what that means is over time, all those nutrients are going to be taken up and they're going to be recycled over and over again. And the cyanobacteria are good at both of those things. So they can take up new nutrients when they come in from surrounding environments, but they're also really good once all those nutrients get taken up and are being recycled in the water column. And so it seems like they have the best of both worlds, but they do best essentially in most systems once those initial nutrients have been taken up and it goes into the recycling phase. That's also tends to be when temperatures are higher in systems as well.
Okay, thank you, Jim. I'm going to go to another question down the line that was upvoted. I'm interested in what seems like an arbitrary difference in activated charcoal and biochar. Is the difference application only, water versus ag? More specifically, can you share how either material might be used to bind nutrients prior to reaching freshwater bodies? Example, remediation and nutrient catchment in streams.
Some of that's semantics. Some of it's so the biochar versus activated carbon. Some of it's in the processing. The processing of the carbon to activate it is a specific chemical process, high temperature heating in low oxygen environments, and it produces the porosity that's present within the activated carbon. It's my understanding the biochar doesn't go through a similar level of processing, so probably it makes the biochar easier to make on site, perhaps. It's probably cheaper. I think either of those could work, and I know that people are trying to test those in like edge of field or other sorts of applications for binding nutrients and things like that. I think that's kind of the application that this question was. Yeah, I mean, they can work. They're not necessarily designed so much, at least activated carbon is not, for phosphorus and nitrogen, but you can functionalize it with other sort of amendments to try to remove phosphorus or nitrogen if that's what you're looking to do. I hope I answered that okay.
Thank you for that. Another question, this one is for Jim. Given your model, would you say that keeping the hypolimnia oxygenated is the most likely approach to manage HABs?
Well, I think it's hard to say what the best approach is, but it's going to be a combination of a lot of things. So the previous question that was asking about BMPs, I think that's an important factor just to limit the nutrients, because that essentially will help you keep the hypolimnia oxygenated. So yeah, I mean, it's a combination of things, but certainly maintaining oxygen in the hypolimnia is a big deal. And I know there were a couple other questions that sort of touch on water quality and methane. So one of the things that I'm really interested in is the fact that methane is actually quite labile. And so the more methane you produce in a system, the more oxygen consumption that occurs in the hypolimnia, because it turns out methane is a reduced carbon. It's as reduced as carbon can be. And so it takes more oxygen to oxidize methane than it does things like glucose or, other sugars. So one of my soapboxes is that an important management strategy for water quality, and kind of touching back on the role of oxygen in the hypolimnia, managing methane is an important way to manage oxygen. And so I think that's something that we need to be thinking about going forward.
Okay, thank you, Jim. I know we've got some questions left. We are at 12:01. So I want to encourage everybody. You've got my screen share is showing both Jim and John's email. So I would encourage you, if your questions didn't get answered today, to follow up with them. I really want to thank them again for their time today, and let you know that if you want to join us next week for a webinar on emerging on-farm research, please do so on the 14th at 2 p.m. Central. So you are getting lots of compliments via clapping hands. So thank you to both of you for your presentations today. And I am going to put the survey in the chat right now. Please take a moment to fill that out as well. So thank you again to everyone. And we hope to see you at an upcoming webinar.